Hip, hip, hooray for DNA. It provides the key to the plans for making everything in you and me.
There are a couple of things we can actually clone. One will be the whole organism. So in this case, we have one organism, like a sheep, and we make a clone of that sheep. That's whole organism cloning. There's something called therapeutic cloning. Therapeutical, that refers to sort of treating disease. That's usually what that is referring to, treating disease. There's therapeutic cloning, and there's gene cloning, where we just clone one gene. Now, we're going to talk about whole organism cloning in this video. This is what we're going to do in this video. But I quickly want to touch on the other two, because you should know a bit about the other two as well. So therapeutic cloning, what that actually is one example with Parkinson's disease and how we use therapeutic cloning to help people with Parkinson's disease. Now, what is Parkinson's disease? Well, Parkinson's disease is actually a disease of the nervous system. Now, the problem with nerve and neurons are obviously part of the nervous system. They make sure that we can send messages. And most cells, almost all cells, do mitosis, right? So if there's a problem with one of the neurons, they will just do mitosis, and there's going to be lots of neurons. But neurons don't do mitosis. So if one of them is damaged, they can't replicate. They stay damaged. They can't replace them. And that's what happens in Parkinson's disease. In Parkinson's disease, your neurons are damaged, and that's why there's often a bit of spasm happening and all that kind of stuff, because the neurons don't get replaced by new neurons. But if we use therapeutic cloning, what we can do is we can grab stem cells, which we can get from embryos or from adults, and we can make clones. We can clone the stem cells, so we go from one stem cell to lots of stem cells, and that's done for cloning. And then these stem cells, they can differentiate. And what was differentiate was if they turn into different types of cells. Stem cells is that original cell that can turn into all different types of cells. The stem cells might turn into, for example, neurons if we want them to turn into neurons. Right? So even though neurons don't do mitosis, we could theoretically make neurons this way. And what we do then is we take these neurons and put them into people with Parkinson's disease, and that will recover their neurons. The neurons that weren't working can be replaced by these new neurons. So we can overcome a problem with neurons that don't do mitosis by making neurons in a lab and then putting them into people. And that's an example of therapeutic cloning, where we clone a certain cell for that cell to be used to treat disease. Gene cloning was just if we have one gene, which is very useful, and the example I could give is a gene that is responsible for making insulin. Remember, some people can't make insulin, so that gene that can make insulin could be quite useful, but we actually put that into bacteria, and that bacteria will make us insulin. But I'll cover them more in a future video, so don't worry about it. I won't go into detail now. But what we can do before we put it into a bacteria is we make lots of copies of it. So we go from one gene to maybe millions of the gene before we put it into bacteria, so we have lots of them, and we can do that for gene cloning. But I won't cover these in too much detail in this video. I'll cover the whole organism cloning in this video. And this is all this picture here. And the example I'll use is nuclear transfer. And that's to do with sheep, but it can also be used for other animals. And the reason why I cover this is because the dot point itself says students will process information from secondary sources to describe a methodology used in cloning. Right? So you have to describe a methodology used in cloning. It says a methodology, so you really only need to remember one. And the one that I'm going to talk about, you don't have to choose this one, but the one I'm going to talk about is nuclear transfer. And in this case, it's done with sheep. So first step is to collect eggs from donor sheep and remove nucleus. That's the first step. So here we have our donor sheep, right? And this donor sheep is not the one that we actually want to clone. This is the one that we want to clone. But what we do is we'll grab her eggs. So we will actually grab her eggs. These are her eggs. But the problem with eggs is the egg itself obviously helps us to make a new embryo and a new baby. But the problem is it has nucleus in it. Her egg has nucleus in it. Now if we were to make a baby with that, the actual offspring won't be a copy, won't be a clone of this one. So what we do is we remove it. So we make sure this one is removed, which goes from this step to this step. Here's been removed. What that allows us to do is allows us to have an egg, which we can then use to produce a embryo. But the egg itself has no nucleus, which is good because we don't want to have the donor's nucleus in there. We want to have the clone's nucleus in there. So that's the first step. We collect an egg from the donor sheep and remove the nucleus. The second step is to collect cells from the target sheep and insert it into the egg. So these cells that we often take, an example of the sheep would be the mammary glands cells. These are where the milk comes out. But overall, it's just important to grab a cell. Remember, all cells in the body have the same identical DNA, right? So it doesn't really matter if it's mammary gland or anything else. They all have the identical DNA. But what we do is we grab the cell from the clone, and now we can put the cell's DNA. So we won't take the whole. What we want is really we want that DNA within the cell. And what we do is we fuse the cell from the target sheep, from the clone sheep. We fuse it with the egg. And why do we do that? Because we want to have the cell DNA inside the egg, because that's what we want to clone. Right, so now we have the egg from the donor with no nucleus, but we have a cell that has a nucleus inside, and we fuse it with that egg to produce a egg that has now the clone's DNA and the clone's nucleus inside. Right, so let the egg fuse with the cell is the third step. So here now we have an egg, which was the original donor egg, but we don't have the donor DNA, we have the clone's DNA inside, which is good because then now it will make an identical copy of the clone. And then what we do is, this happens in the lab, we let the egg become a small embryo, right? So let the egg develop into the embryo, that's stage 4, so stage 4 is here. This was stage 1, this was stage 2, this was stage 3, stage 4, and then once we've got the embryo, we put it into the donor. So this is the donor sheep. Right? So this is the sheep that donated her eggs. And what we do is we grab the embryo and put it inside of her. So place embryo into donor sheep and wait till the new one is born. What happens when the new one is born? The mom that gave birth to this one had a black head, but the actual baby had a white head. And the reason why is because this baby has nothing to do with the donor. It has all to do with the clone. This is a clone. The baby is a clone of this one here, and that means her head is white because their mom, or the clone's original one, is also white. Right? So we just use the donor just to deliver it, but genetically wise, that sheep is related to the clone itself. And this was the process of how we can do that. And this was actually, I'm pretty sure this was how the Dolly the sheep was made. And the Dolly the sheep was the first cloned animal. I think that was 1997. Right, so for this dot point, what you should remember is you should remember a way that we can make, that is using cloning. The example I gave is nuclear transfer. You should remember the steps and just the gist of it, right? So that we have this donor sheep and the sheep that we want to clone. And we remove the DNA from the donor sheep. We put the DNA from the clone sheep into the egg, then make sure it becomes an embryo, and then once it's an embryo, we stick it back in the donor, and it will give birth to it a couple of months later, but the actual baby won't be related to the donor, it'll be related to the clone itself, because it is a clone of the original white sheep here, in this example. Okay? I hope that was useful.
Thank you for watching.